TOXBANK Data Warehouse has been established to house all the data and experimental protocols generated as part of the SURAT1 cluster. The first step to take a look at the data and protocols is to log in. So we'll go to the services.toxbank.net and fill in the username and password. Now the data is uploaded in a format called the ISATAB Universal Data Exchange format. And there's a tool available from the help. Here you can see installation of the ISA, it's called ISA Creator. There are 2 downloads. If you wanted to take a look at the ISATAB data in these formats, first you have to download and install this application. If we go back to the search page, and we search for a specific investigation record. It's going to search through the TOXBANK Data Warehouse to identify that record. And here, as you can see, this is the record. And if we single click on the link, it's going to take us to the full details. So here's an abstract of the record. Here's some keywords, and you can also see there's a red link here. If you click on this button, you can download the individual data files. This would include the raw data as well as processed data. Going back to this, we can also see an investigation document, and this is the document that's in the ISATAB Universal Data Exchange format. So let's go ahead and save this document, because what this will describe is fully the experiment, both the experimental factors, the processing of the data, and as well as how the data is generated. So let's call this and we're just going to save this to a folder. And if you look at the link here at the top, it's on my C drive. It's where you installed the ISA Creator, and in the folder called ISATAB Files, we're going to save that zip, and then we're going to unzip it in this folder. So let's go ahead and save that folder. The download has been complete. So now you can see here is that specific folder. And I'm just going to extract into that folder this archive. So once that has then been extracted, what we can then do is we can start up the program that we just downloaded and installed called ISA Creator. And we're going to select the ISA Creator software. And you do have to log into this application. And select the TOXBANK configuration. So let's load an existing ISATAB file, and as part of your download you should see that folder reference data for TOXBANK. And the ISATAB file is organized according to the 3 levels of ISA. I stands for investigation, S study, A assay. So the top level reports information on the investigation, such as an identifier, a title, a description, any publications, and a contact for this investigation. The S stands for study. So within an investigation you could have more than one study, and a study is generally around a particular biological target or a cell, and the study may include 1 or more assays. That's the A in ISATAB. So in this case, this is a study. You can see the funding sources. It had 1 assay, and this was a transcriptomics profiling DNA microarray assay. And if you look at the study, there were 3 experimental factors: compound, dose, time point, which have been used in the study. And you can also see where individual steps of the study, there is a protocol that's been uploaded to the TOXBANK Data Warehouse. So let's look at the study in a little bit more detail. So the study was performed, and as you can see from this this table, just gonna we can look to see the organism, the sex, the strain, the characteristics of this, what has been measured. So these are hepatocytes. It's an in vitro assay. So these are all characteristics of the assay. There was 2 replicates for each test, and here you can see information, for example, on the study factors. So here are the compounds, so paracetamol, aflatoxin, carbon tetrachloride. These are all the compounds that were tested. Here you can see that the first 2 entries were negative controls, and if we look at the dose and the dose unit, they were 0 dose. Here you can see the dose levels that were tested. These were the low, the control, the low, medium, and high dose levels. And also the time points were at 2 hours, 8 hours, 24 hours. So each one of these rows then is assigned a unique number, and this is then used in the assays, and this can be looked at in more detail. So if we go to the step down, so we performed a single transcript omics assay, and here you can see that sample name. So you understand from this the source, what the treatments were, what the information used to generate the sample. And then there's a series of steps. So there's an extraction protocol that creates a new extract name. There's a protocol for the labeling. Here you can see the particular label that was used, biotin. The protocol for the hybridization. And these are just a series of steps that are performed. We had the scanning and data acquisition. Then this resulted in a series of raw data files, in this case cell files. Here you can see the array data files, and here are the individual cell files. And these are all available on the TOXBANK warehouse. So if I click on this download, there's a convenient link to those raw files. So here you can see these are these individual raw files if you wanted to download them individually. So this data was then normalized, and the protocol is describing that. Again, you give it a name, and then this raw data, derived data file, again has been loaded to an FTP server. Here is the link to it, but again you can see the GC derived data file. It's available for download from the warehouse. Here it is, the derived data file. You can download it directly. Finally, all this information was then processed, and you can see, for example, in the data transformation that multiple cell files, were analyzed. So if we look at APAP, acetaminophen, 2 hours low versus control, there's in fact 4 entries here that correspond to this control. And this is then used to calculate the derived matrix. And the derived matrix is assigned in a standardized way. And again, this can be downloaded from the data warehouse. Here is the derived data matrix. You can also see it's a file name, which is actually inside the folder that we unzipped with the full ISA tab. Now this has been carefully developed, and if we take a look, I'm just going to open up Excel and open up that specific file. So here we're going to navigate to the reference set, and I'm going to go to this file and import it into Excel. And what this is now showing is the processed data results. So here you can see the first few columns are identifications for the genes. Then there's a series of processed data file columns. So here you can see a column for FC. This stands for the log2 fold change. Here you can see APAP 2 hour low versus control. This is the name that appears in the ISA tab. Here the APAP 2 hour low versus control. This is the processed field, so you can see for this particular one the fold change, the up or down fold changes. However, you can go back to the original data to understand a complete chain of processing that took from the experimental design through to the data generation of the cell files, for the processing of the cell files to get to this point. So there's a full audit trail of how you got to this data. But this is then information that can be then used in an integrated analysis or pathway enrichment and used to process and understand the data in more detail. So here you can see there are control, there's fold changes. You'll also see things such as p-values and q-values for the values, which are the p-values are the raw p-values, and the q is the adjusted p-values for the false discovery rate. So that summarizes how you can search this data warehouse for data, download the ISA tab, and understand exactly how the experiment was performed and how the processing of the data was achieved.